stimulated by this complex. So these papers are my first author papers in Columbia. I use these papers to get a job. Then these papers are published in my lab in China. Okay. So later, I switched to a new field, because our lab belonged to the state laboratory of developmental biology. They said, okay, you are here doing something on development. So I chose one of the tough projects on brain development. So it works. I survived. So that's one of the major reason I went back to China, because in US, if you switch to a field, that's kind of suicide, right? So in China, I survived well. Okay, I'll tell you, we are working well on this. Okay, so one of the things I'm going to talk about today is WDR62. So why we study that? Because 5 years ago, it has been found mutation of this gene will cause microcephaly. Okay, and so it's represented by the small brain, and also those patients are, one is retarded, expected, and another thing unexpected is those patients kind of aggressive, and some of them have autistic, behavior. That means maybe related to autism. Okay, and WS62 is the second most important gene ever found for microcephaly. So the first paper we published on that is we use the electroporation to knock down WS62. Okay, so first thing you can say is affect migration, right? So the cells cannot migrate up to the cortex. And if you notice here, it's interesting. We also lost the stem cells. And this is due to its interaction with junk to regulate the differentiation. Not me, right? Okay. Regulate the differentiation of the neural stem cells, cause the premature differentiation. So now I will talk about the unpublished data. Okay. So we make a knockout, a conditional knockout. Okay. You can see the brain is much smaller, right? And we also did the behavior, use different kind of behavior to test their learning and memory. They are retarded, as expected. Okay. But this will be interesting. We also test, social interaction. Okay, they have trouble to communicate with each other. Okay, and so this is a very dramatic phenotype. This is heterozygous. Okay, if you look at the papers, the publications, okay, the most dramatic phenotype is like this. Okay, there are 3 Nature papers published on different genes that has this kind of phenotype, but that's homozygous. This is heterozygous, more related to disease, and so here is the phenotype for homozygous. They fight each other, okay? They hate each other. Wherever they meet, they fight, and they fight till death. Okay? So that means one thing interesting is we screened autism kids, okay? One case showed de novo mutation of this gene. That's very interesting. So that means W62 may be associated with autism. If we look at the embryonic stage of the mice, you will see the cortex is much thinner, right? And especially this layer 4 neural stem cells, and also the enlarged lateral ventricle in the brain. Right? Then we use different markers to label different layers of brain. Okay, our brain has 6 layers, and each layer is thinner. And if we look at the SOX2, that's labeled neural stem cells, they are thinner too. So the question is why they are thinner, okay? Why we lost those neural stem cells? So we culture neural progenitor cells from those knockout mice, and they form much less neural spheres, okay? And even they form the neural sphere, the size is much smaller. That means it affects neural stem cell proliferation. Right? And why it affects that? We look at in the brain the phosphohydrin 3. That's a marker for cells that's in the M phase. Okay? You can tell much less cells in the M phase than in the control wild type. Okay? And then we use KSC7. That's labeled all the cells undergoing cell proliferation. Okay? and it's much less here. And we also use BRDU that's labeled S phase cells. Okay, if you combine this together, you can tell the cell cycle exert index. What does that mean? That means how many of the cells differentiated into neurons. We know neurons are in G0 phase. So if this increase, that means more cells has differentiated. So we get the increase of the cell cycle exert index. That supports the conclusion that they get premature differentiation. That's why we lost these progenitor cells. And so what's the cellular mechanism? Okay, you can see we got a lot of those, abnormal spinal cord, spindle pool. Okay, that means the cell cycle get affected, is affected the spindle pool. Okay, if you look at the division of the neuron stem cells, they divide in control is this way. Okay, that means expansion of neuron stem cells. If you expand it this way, that means one cell is still neuron stem cells, while the other will be differentiated. So that's why we lost the neuron stem cells. Okay, so we have shown before WRX2 interact with JAK1 to regulate neuron stem cell differentiation, right? The question is, what's the upstream here? Okay, we try to find out which is upstream gene. So we use siRNA library screening for all MKK case, only 20 of them. So after screening 5 or 6 of them, we found one that has exactly the same kind of phenotype as knockdown of WRX2. Okay, defect in migration, loss of neuron stem cells. Okay, so we can change the model to this way. Okay, it's MEKK3. Then we get the knockout mice and study what's wrong in MEKK3 knockout mice. So we get very similar phenotype. The loss of neuron stem cells labeled by PEC6, SOX2. It also has much less phosphohistone 3 cells here. And so whether WRX2 is indeed related to JNK pathway. Okay, so we look at the knockout mice, and the JNK activity is down-regulated a lot, lost about 2/3 of the activity. Okay, so it's related. Then we make a transgenic JNK1 knockout, transgenic JNK1 mice. Okay, why we make that? Because people made that before. Overexpressed in the whole body is kill the mice. Okay, you won't get it because if you active JNK for long, you kill the cell. So we have to use a inducible JNK1, transgenic JNK1. Then we use this JNK1, and first we look at the knockout, the phenotype in knockout. The phenotype is very similar. Okay, thinner of the brain and the enlarged lateral ventricle, and increase of cell cycle index. Okay, is also cause premature differentiation. Okay, then we make a transgenic mice and cross that with WRX2 knockout mice. Okay, and you can see the small brain size can be rescued, and as well as the enlarged lateral ventricle can also be rescued very well. Okay, so that indicates WRX2 regulates JNK pathway, and the JNK1 can rescue WRX2. And then we look at the stem cells, whether they can be rescued or not. They can be rescued by JNK1. Okay, and as well as cell cycle exert index, the premature differentiation can be rescued as well. Okay, so that's genetics. Then my lab is a single transduction lab, so we did a lot of biochem work. And so the first thing interesting is if we overexpress WRX2 and MEK3 together, they are much more than expressed alone. WRX2 and MEK3. And then we use JNK1, we can see JNK1 can stabilize WRX2. So why is the case? So we did a mass spec and found a dozen different phosphoryl sites in WRX2. And I'll talk about these three sites, okay, four sites. So when we mutate these three sites as a system of WRC2, you can see it become very unstable. Okay, compare this with this. And when we look at the knockout mice, the cortex, the WRC2 level is much less in the wild type. This indicates MKK3 can active JAMP1 to cause the phosphorylation of WRC2 and stabilize it, right? And as I tell you, activation of JAMP pathway has to be controlled. You cannot activate it all the time. So there needs to be a negative control factor, okay? Then we look at this point. And it's interesting is this point is also important because it's bound to ABW7. Okay, I want to give you a lot of biochem study. Okay, just give you a result. So this point, this site, when we make it to alanine mutation, the proteins become very stable compared with wild type. And when we make a mimic mutation, it becomes very unstable. Okay, so why is that the case? Because ABW7 is an E3 ligase that's degraded the protein. Okay, then we confirm that is mimic mutation. You can see a lot of ubiquitination, and in the alanine mutation, much less ubiquitination. Right? Okay, so ABW7 degraded WDR62. So and also in the ABW7 knockout mice, we can see WDR62 is much more than that in the control. Okay. So the genetics also support our biochem data. So I won't give you too much detail, but just give you the conclusion. We did a lot of cell cycle study as well. So the WDR62 interact with those components to organize this complex. And MKK3 can active JAMP1 to lead to the phosphorylation of these three sites. That's at SG2 phase. Okay, when the cell cycle gets to M phase, this site also gets phosphorylated by JAMP1, and that can recruit the E3 ligase ABW7, which lead to the degradation of WR62. Okay, that's in M phase. And together, through this mechanism, is regular cell division as well as non-stem cell maintenance, the self-renew of non-stem cells. So when we knock out WR62, the JAMP cannot be activated. Okay, so that would lead to the asymmetric division of neural stem cells, loss of the neural stem cells, small brain, and the defect in learning and memory, as well as autism behaviors. So, okay. WRC2 here, we have done a lot of work, in TGF signaling pathway because years ago, 6 years ago, there are almost no publication of TGF signaling in brain development. Okay, although people have done that in, embryonic stem cells, but not in neuron, not in brain, not in vivo. Okay, so we did have found two complexes. Okay, so this complex regulate neuronal migration. Okay, this complex regulate the differentiation of neural stem cells. So we also found in this branch, BMP can active SMITE that can inhibit CRAM2. That is a schizophrenia associated gene. And we have another paper, published on schizophrenia. So basically, that's it. Thank you for your patience. So today, most of the unpublished work is done by this lady called Dan Xu, and this our big family of the lab. So with a boyfriend, girlfriend, husband, or wife. So
thank you.
Okay.
Well, this is very interesting talk. Open for questions.
Yes.
So with the JAMP rescue,
Right.
it didn't rescue the behavior, did it?
Yeah, that's a good question. So.
Because, it's not regulated. You grew more cells, but did they act normal?
Yeah, the size is become normal. Normal. We haven't done the behavior test. You know, the gene is
But it's such dramatic behavior. What did they look like in their cage?
wow, that's you know that's pain in the neck. You know, so you know when my student did the experiment, so each group will have at least a dozen, right? So after the experiment done, halfway, a lot of mice got killed because they kill each other.
Okay.
Then the students think, okay, maybe I'll put that together with wild type. So one knockout, one wild type. Then do the behavior again. So it take 3 months to do that, right? And then the behavior become abnormal. You know why? Because the knockout beat the wild type.
Right.
The wild type become depressed.
Okay.
So eventually we have to culture those babies alone. So the experiment become very expensive.
So this WDR62, is it only when you knock it out, except the neuronal stem cells? What is the impact on other stem cells? I mean, is it,
A lot, I can tell you a lot. So we, you know, the homozygous become sterile. So it affect the oogenesis, affect spermatogenesis. So that's why we have to use conditional knockout to do all these experiments.
all right?
Yeah.
Very interesting. Thank you very much.
Okay.